Good morning. My name is Carrie Mounty, and I am from Trilogy Analytical Laboratory in Washington, Missouri in the U.S. We want to welcome you to this virtual mycotoxin workshop today. And on behalf of Trilogy, I certainly appreciate the opportunity to share some mycotoxin information about mycotoxins in general, as well as some tips and techniques that will help build a solid testing program in your facilities. First, I would like to take a moment and give you a little background on Trilogy. Our company was formed in 1999, and since our inception, we have had a solid emphasis in mycotoxin testing services and also offering products that support toxin testing. In the U.S., we handle a high volume of mycotoxin analyses, and we run between 200,000 and 250,000 analyses annually. We have our ISO 17025 and our ISO 17034 accreditation, and our facility also holds an ISO 9001 certification. We do have several facilities around the world, and that has allowed us to broaden our range of services and activities and our products. In addition to the U.S., we have facilities in Latin America and Europe, and a 2021 expansion opening in Ghana. As we all know, mycotoxin testing is a critical part of global trade. And unfortunately, this gets more complicated because many countries have different mycotoxin regulations. Imports that don't meet in-country standards, as well as any exports that are not within the recipient's specifications, have a financial impact that can be a huge burden for both importers and exporters. Additionally, the imported commodities headed for both human and animal consumption must be taken into consideration health consequences that are associated with those mycotoxins. Mycotoxins are chemical compounds produced by molds. There's actually hundreds of mycotoxins. One such toxin that we all know about is penicillin. Sometimes at the lab, we will get asked how to kill a mycotoxin. This is hard to think about sometimes because we consider that the mold can actually be killed, but the toxin, like any other compound, such as, say, salt or sugar, can't be killed. Mycotoxins have been around since molds began growing. In fact, one of the plagues was thought to have been a mycotoxin called ergot. So, where exactly do these toxins come from? Well, different weather conditions produce different molds, and each mold produces different mycotoxins. So weather conditions typically are associated with different toxins. For instance, the molds that produce deoxynivalenol, which is also known as DON, or vomitoxin, typically prefer wetter, cooler conditions. In addition, plants are more susceptible to mold invasion during specific times in their growth and development. Any broken kernel, damage from insects, immature kernels, etc., will allow entryway for molds. And once those molds are in, they have a food source and they can then produce toxins. This type of contamination happens in the field prior to harvest. However, improper storage and transportation can also increase molds and mycotoxin production dramatically. Mycotoxins therefore enter the food chain both pre-harvest and post-harvest. Managing both points of the process is essential for solid risk management. I mentioned there are hundreds of mycotoxins. However, only a few are what we would call common mycotoxins with which we should be concerned. Mycotoxins are associated with weather conditions, but they can also be associated with specific commodities. The chart below shows the toxin and commodity associations as well as the general weather conditions associated with each mycotoxin. You will notice that corn is associated with all of the toxins. Corn has a wide production area and it is grown in many climate conditions. Wheat, on the other hand, has a more limited toxin association. The most common toxins associated with wheat are DON, zearalenone, and ochratoxin. One thing to remember, the processed products associated with the main commodity may also contain mycotoxins. In fact, many processes concentrate the mycotoxins. For instance, in wheat, the wheat mids, which are also known as the middlings, are that outer portion of the wheat kernel, typically have the highest concentration of DON, whereas the innermost part of the wheat kernel has the least amount of contamination. There are some physical signs of mold damage. It is important to remember, however, that these signs are only indicators that mold damage has occurred. It is not an indication of toxin contamination or toxin concentration. This slide shows this fact in detail. On the right-hand portion of the slide, the wheat kernels at the top show healthy wheat kernels. The kernels at the bottom, however, show the pink-tipped kernels as well as some tombstone kernels. These two characteristics, the pink tips and the tombstone kernels, are physical signs of mold damage. The tombstone kernels occur when mold has entered the kernel and consumed the carbohydrate source in the middle. This results in a very lightweight, hollow kernel that has been named a tombstone kernel. The corn kernels on the left side of the slide represent some research on single kernel corn we performed at Trilogy. The very damaged kernel was analyzed and found to have 0.6 ppm or 600 parts per billion DON. Right next to it, the yellower, healthier looking kernel was analyzed individually and found to have 7 ppm or 7,000 parts per billion. So the healthier looking kernel had way more DON than the kernel that looked much more damaged. The physical characteristics of these two kernels do not correspond at all to the toxin concentrations we found. The important part of this slide is to understand that only testing can confirm the type and the amount of toxins that are present in a sample. While we are emphasizing that only testing can truly confirm toxin presence and concentration, there are some physical guidelines that can be evaluated to determine if mold damage was a potential that could lead to possible toxin contamination. Many purchasing points will put out some guidelines in place to limit acceptance of lots of wheat with increased DON potential. For instance, perhaps limiting scabby wheat, which is a term for the tombstone or pink kernels that you see in wheat, that those not exceed 2 to 3% of the total kernels evaluated. Also, low test weights per bushel are also an indicator that molds may have been present. Remember, those molds had to have a food source, and those come from the kernels. If the growing season where the wheat originated was cool and wet, this is yet another indicator that the toxins could be problematic. And remember, if the point of purchase brings in wheat mids, those are always suspected as having the highest toxin concentration. As many of you have either already implemented a testing program or perhaps are contemplating implementing a testing program, I want to take this time to highlight some of the testing challenges you may face, regardless of how you are performing the actual test method. First, let's look at the bar graph. This bar chart represents the total variability of a mycotoxin sample. The bottom blue portion of the graph represents the sampling portion of the process. Sampling includes how the sample is actually procured. There are many sampling diagrams available for everything from trucks, rail cars, and silos to feed bags, bunkers, or ship containers. The method that these vessels are sampled is important. As we have been talking, mycotoxin contamination is very inhomogeneous, so it takes some work to get a representative sample. How many probes will be taken? How much sample will be taken in each probe? What happens to the samples when they are compiled? How will these probes be compiled? All of these components of sampling are important. The red portion of the graph shows the variability that the actual preparation of the sample adds to the total result. This covers everything that is done to the sample after it's collected. That typically takes place in your laboratory. Things that affect this part of the process include how finely the sample is ground. Is every kernel sampled? How much analytical will be extracted? What extraction solvent is the one to use? How long will the sample be extracted, and how? By hand shaking? By a blender? These are all critical parts of sample preparation that lead to the total variability of the answer. The green portion, which you can barely see, is the variability that actually comes from the test method. Some methods, such as LCMS-MS, may have an overall lower variability in the method than perhaps a rapid test. However, you can see that this portion of the process is not what makes up most of the variability. In summary, sampling, or how you get the sample, makes up about 61% of the total variability. Sample prep, or what you do in the lab to prepare the sample, makes up about 35% of the variability. And the actual test method, only about 3% of the variability. These numbers vary by specific toxin, what the actual toxin concentration is, and the commodity, but you get the general idea. To further show the sampling part of this variability, the table above the graph shows an actual study done with corn that contained approximately 20 parts per billion aflatoxin. The left column of the table indicates how much sample was probed from the truck. As you can see, the truck was probed numerous times. The right column shows the variability when multiple samples from that probe were analyzed. As you can see, the bigger the probe, the smaller the range of results. This shows that a larger probe when sampling vessels is important. The smallest sample probe resulted in a variability from less than 1 to 46.9 ppb. The results you get will be much better with a properly obtained sample. What we call the ppb challenge is the next thing we'll discuss. Mycotoxins are quantitated at the ppm, parts per million, and ppb, parts per billion level. Think about the small amount of material we're looking for. A ppb is 1 second in 32 years. There's some other examples, but that one is just really definitive of what a small amount we're quantitating. Maybe it seems like we're looking for a needle in a haystack, but when we recognize that these parameters affect our total result, it will help us get the best sample possible. Next, and we have hit on this challenge already in the previous slides, is the distribution challenge. Mycotoxins are not like protein, fat, or fiber, or moisture. A lot of those parameters we understand when we analyze commodities. Because mycotoxin contaminations happen where insects have damaged kernels or we find broken kernels, it's very inhomogeneous. Only 1 out of every 200 kernels of corn may be contaminated. If we're looking at cottonseed, only 18 seeds out of 150 were contaminated. You get the idea. You have to sample a lot of grain kernels. With this background knowledge, let's look at some sample prep videos to help explain this concept even further. Sample splitting is a very common practice, and it works well for the analysis of something like moisture. But let's look at what happens when whole grain is subsampled for mycotoxin testing. The picture below shows mycotoxin contaminated kernels in among healthy corn kernels. Let's see what happens when we riffle this. This is a standard riffler, much like you would see sample splitters. We're going to take one contaminated kernel and place it in 199. As the sample split takes place, there's no way that the sample on each side is going to have an equal amount of mycotoxin. This tray will clearly have more toxin than the total sample. The next tray will have no mycotoxin. So one of those samples will be a false negative and the other will be a high positive. Let's see what happens when we do 5 kernels in 925. We'll take those fluorescent kernels and mix them around before we split. These splitters are very similar to what you might find in a grain facility. Let's look at the results. In this container, we're going to see 1, 2, 3, 4 kernels representing kernels with mycotoxin contamination. This means that in the other split, there is only 1 kernel with mycotoxin contamination. This represents a low positive and a very high positive. Now let's look and see what happens when we finally grind the material. The material that we're grinding is the 5 kernels in 925. We're grinding them to a fine consistency and we're going to subsample. As we can see in this diagram, all of the fluorescent particles of that piece of corn or those pieces of corn are evenly distributed. Let's talk about that final result. What goes into the accuracy of that number that you are going to make your decisions on. Let's take an example of a wheat sample that was found to contain 3.2 parts per million DON. This level may be a result that is on the borderline of being a level that is either accepted or rejected at your point of purchase. Don't forget, this number represents a financial decision. If this number is too high for some reason, perhaps you are rejecting a product that you should accept. And on the other hand, if this number is too low, perhaps you are going to incorporate a wheat that has a higher level of DON into a finished product and be forced to either perhaps issue a recall or deal with some other out-of-spec product that you're going to have to make decisions on. This overview of result variability shows areas that contribute to what we call kind of the wiggle in our results. We have talked at length about some of those items, but as you go forward, remember to take into consideration things like technician training. Were they all trained well, and is everyone doing the same procedure every time? The testing frequency. How often should you test? What about the method suitability? A lot of times, a method that works well on wheat won't work well on, say, something like spices. And what about the particle size of your analytical sample? Remember, smaller grind size is going to give you more particles. Confirmation of sample results. It's always a good idea to take a certain number of results and confirm them to make sure that you can repeat that result. What about equipment calibration and performance? Do you have some ideas on how to routinely check those parameters? These are only a few of the things that make up an accurate result. Your system will have items that are not listed here. As we wind up the section on general things that will help establish a solid testing program, remember, a testing program will help mitigate some of the risk inherent in mycotoxin testing and provide you with the best data to make those important decisions. As a review, remember, with so many things about mycotoxins that are out of our control, things like the weather, our suppliers, the commodity you have to work with, it becomes even more important to control all of the things that are in our control. Things like how we do sample prep, how we train our staff, how we confirm our results, etc. Next, educate yourself and your staff about parameters that are out of your control. Can you watch weather patterns and determine if problems are a possibility? Also, look closely at your commodities and know what toxins are most likely to be problematic. Set some guidelines for acceptance criteria on physical characteristics, perhaps like bushel weight. Finally, focus on simple but important elements. Molds thrive in moisture conditions above 14%. Dry products before storage and ensure that no hot spots of moisture are being formed. Make sure transportation time, temperature, and moisture is not compounding the problem. Know the requirements of your clients and what toxin levels are critical to them. Don't forget, perhaps human consumption products may not have the same requirements as those for animal feed. Or maybe wheat midds that are used simply as a fiber source may be able to have a higher level of DON. All of these things are important components of a solid testing program. Reviewing these items ensure that you are setting up a system that has the final result in mind from the very beginning. Now, let's take some time to talk about the actual testing options. It's important to choose a rapid test method that accommodates your specific needs. You need to look at things like the physical space you have to do the testing. What type of a staff do you have that will perform the test? Evaluating these specifics will lead you to make the correct choice on which test fits your needs the best. There are a variety of different methods. Let's talk a little bit about two rapid tests. First, we'll talk about the lateral flow methods. These methods use a small plastic device that can be read as a single sample. These devices have many benefits. First of all, they are very rapid. Many times, it's only minutes from the extraction of the sample to the final quantitation. They are extremely easy to use. There can be on-site training or actually web training can be used because the methods are easily learned. There's no dedicated expensive equipment. Typically, what is used are pipetters and some type of a reader, and in many instances, a cell phone can be used for quantitation. These methods are nice because you can run a single sample. There's no need to use unnecessary sample wells if you're only running one sample. The calibration curve is already programmed into the reader or the phone. These tests are great for the point of purchase places when they receive many lots of materials at different times. The next type of rapid test also has a lot of benefits in a little bit different situation. ELISA tests, like the picture on the right of this slide, are great for running many samples together. Many times, depending on the well size, 20+ samples can be completed in one run. Another very nice thing about this technology is if you are in a facility that has this type of sample load, automation is available. Large numbers of samples can be run with little intervention because the robotics do the pipetting and the instrument will let you know when the results are available. Additionally, typically with these ELISA formats, data is downloaded to a computer, so more complex data manipulation is available for those facilities that need this type of testing. Regardless of what type of methodology you choose, testing for mycotoxins is here to stay. Currently, mycotoxin testing is performed by many companies in a lot of countries around the world. Many of those countries already have regulations in place, and other countries are implementing those new regulations for things that they import. The methodology around the world varies from a very rapid qualitative test to very sophisticated quantitative reference methods that are performed typically just in laboratories. There are also trends for lower and lower detection limits. And with global trade seeing more commodities going into new countries and new products being developed, mycotoxin testing is on the rise. In the next presentation, you will see a rapid mycotoxin test and the ease at which it is performed. Selecting a rapid test is a critical part of your total program. At Trilogy, we developed our quality system with analytical methods, but we realized that we also needed additional tools to round out our total quality system. We developed tools for our internal use, and soon we were being asked by our analytical clients to provide these tools to them so they may use them with their rapid on-site methods. Now, our quality tools include reference standards, known quality control materials for training and run acceptance, a laboratory shaker, purification columns, proficiency samples, just to name a few of the tools. We pride ourselves in helping other companies set up the best quality system for their mycotoxin testing. In our mind, helping others to obtain accurate mycotoxin results helps everyone in the end. If we can help in any way, please don't hesitate to contact us for more information on any of the topics we reviewed today. Thanks so much for your attention. We look forward to any of your feedback or questions.